on the topic of partnering to accelerate discovery in EHE. And our first speaker is going to be from Margaret Cancer Center, Catherine Weadick, who's going to talk on the cancer or the CanSaRC and ProCare EHE insights from Canadian research. Welcome aboard, and please begin when you're ready.
Good morning, everybody. Dr. Catherine Weadick is my name, and I'm a clinical research fellow working in Princess Margaret Cancer Center and Mount Sinai Health. And I'm going to present to you today. I would just like to thank the committee for giving me the opportunity to present. So I'm going to talk to you about our project, ProCare EHE and CanSaRCC, and I'm presenting on behalf of all of my co-authors. So what is ProCare EHE? ProCare EHE is a multi-pronged project of Canadian research in EHE. So it was funded by a grant from the Sarcoma Cancer Foundation of Canada. The clinical lead is Dr. Albie Rooney-Razik, and the scientific lead is Dr. Kristianne Allen, and she runs a lab in the University of Toronto. We collaborate with doctors locally here in Toronto, such as Dr. Gupta and Dr. Salu, and also those internationally, such as Dr. Staticacci in Milan and Professor Jones in London. And I'm going to talk to you a little bit about their input as we go through the presentation. So we have four aims or four prongs of this project. So the first is the Pan-Canada EHE data curation. So what does this mean? We were hoping to create a database of EHE patients in Canada who are currently living with EHE, and that is what the bulk of this presentation is going to be about. We then have three further aims where we're looking to create a prospective international database, to create tumours of EHE in the lab, and also to test treatments in the lab on EHE tumours, and I'm going to tell you a little bit about those towards the end. So, as we all know, EHE is an ultra-rare sarcoma, so I won't get into that, but the reason that we wanted to do this project is because of the rarity. We wanted a study that was going to describe the presentation, the management, and the outcome of EHE patients in Canada, and we did this through CanSaRCC. So what is CanSaRCC? So CanSaRCC is a Canadian research collaboration that looks into sarcoma nationally. So it's a group of Canadian researchers and doctors who are interested in sarcoma, and they enter their patients into a database. And this was founded by Dr. Gupta here in Toronto, and was how we were able to do this project. So not only does it look into EHE, it looks into loads of different sarcomas, and a lot of projects have come from that. So how did we do this project? The patient's data was entered into this CanSaRCC database, and we included people of all ages who had a documented diagnosis of EHE, who were diagnosed between November of 1993 to January of 2024. So everybody's demographics, such as their age and their site of cancer when it was diagnosed, and the treatments that they had, were collected using a specific form and then entered into the database. And a response assessment was performed by the doctors locally. So what does that mean? That means that when we looked at the scans to see how the tumour had responded to treatment, that was done by doctors in the individual hospitals locally. So this is what CanSaRCC was able to achieve. So we were able to have people from 10 different hospitals across Canada to be included in this project. So this includes both paediatric, so children's hospitals, and adult hospitals across nearly 31 years. So our results. So we were able to include 198 patients with a diagnosis of EHE in this project, and it was evenly split between men and women with 99 of each. The median age, so the average age of patients included in this project, were 51, and the youngest was 9 and the oldest was 86. So a very broad range of people were included in this project. So when we look at their primary site of diagnosis, so that means the site where the cancer had started at the time of diagnosis, the majority of patients had EHE start in their liver, with 56 patients. And this was then followed by 31 in the extremity, which is the arms and legs, then followed by lung at 27. And the remainder of patients had other sites at diagnosis. When we look at disease presentation, so how the EHE was found, The majority of patients at 63 presented with symptoms of a pleural effusion. So that is fluid around their lungs. So that could be shortness of breath or a cough and sometimes pain. Next, 61% patients presented incidentally. So that means that it was found by accident if they were having scans for something else, or maybe they had been in a car accident and they had a pain, or they showed a lump when they were in the hospital for another reason, and that was how it was found. And then this was then followed by 25% of patients presented with ascites. So ascites is when you have fluid in your belly, and sometimes that can present with swelling, pain, or a change in your bowel habit. And of these patients, 94, so nearly half of the patients had metastatic disease at diagnosis. So this means that their cancer had spread beyond the site of their primary site, so beyond just the lung or the liver. It had gone to other places, or perhaps they had multiple spots in their lung or their liver. And then we look at the molecular testing. So as we know, to diagnose EHE, patients need to have one of the two fusions. So we look to see who of our patients had the testing done. So of our 198 patients, 54% of patients had this testing done. Now, we should keep in mind that this database goes back to 1993. So that's 27.3% of patients. And of those patients that were tested, nearly 80% were positive for either of these fusions. And we do see that since 2015, that there has been an increase in the molecular testing. So this has increased to 48.5%, which is a positive finding. So then when we look at the treatment that these patients received, so the majority of patients underwent curative surgery. This was then followed by systemic therapy. So systemic therapy is either a tablet or treatment that is given through a needle in your arm. And when we look at what treatments were given, so in the first line setting, so this means that the first systemic treatment that patients received, the majority of patients received sirolimus. This is an mTOR inhibitor. This was then followed by another tablet called pazopanib, and then followed by chemotherapy, such as paclitaxel or doxorubicin. And when we look at the second line, so second line is when the first line treatment has stopped working and the EHE has started to grow, or perhaps the person wasn't tolerating the treatment and needed a second treatment option. And when we look at these treatments, it looks like pazopanib was the most common treatment, then followed by the chemotherapy doxorubicin, and followed by sirolimus. 28% of patients had radiation therapy, and liver transplant was performed in 8 of our patients. And this spans from 2004 to 2019. And then when we look at survival, so we look at how long people lived from the time of their diagnosis to the time that they passed away, and also factors that could have influenced this outcome. So we use things called Kaplan-Meier curves. So these are these graphs that you see to document survival. So on average, patients were followed up for 4.5 years. And this ranged from 1 month to 28.5 years. And when we look at the average survival, so what we call the median overall survival of someone diagnosed with EHE, this was 8.6 years. So this means that the average person had 8.6 years from the time that their EHE was diagnosed to the time that they passed away from either EHE or from another cause. But we should keep in mind that this is a median number. So that means that 50% of people did worse than this, but 50% of people did better than this. And this table at the bottom, table 3, this is multivariate analysis. So what this means is we look at different factors that is associated with people having a poor survival. So this found that people who had a pleural effusion or ascites, so that is fluid in their lung or in their belly, those that had EHE that started in their lung, and those that had metastatic disease were found to have a poorer overall survival. So that meant that they were more likely to have a shorter time with their diagnosis of EHE than those without these factors. So our conclusion from this first aim, so what has this aim taught us, or what have we been able to achieve with this? So we were able to describe the largest series of EHE patients in Canada to date, with 198 patients, which span 31 years. And we have since published this data, and it is the largest series published with detailed treatments that patients received and also how they responded to this treatment. It highlights that people with pleural effusions and ascites and primary lung disease had poorer outcomes. It demonstrates the limited uptake of molecular testing. However, this has improved in recent years. The underutilization of liver transplant, and also poor survival outcomes with limited efficacious treatments for people with EHE. So what have we done with this? We advocate for routine molecular testing, for national guidelines surrounding the role of liver transplant, and also for a prospective international database. So that means that data going forward for people to be enrolled as they're diagnosed with EHE internationally. And some of the other prongs do help with this. So we're going to discuss this now. So that was aim 1. So then our other 3 aims. So our aim 2 is a commitment to a multi-institutional collaboration. So we are collaborating through CTOS and a platform called PUSH to develop an international database of patients with EHE. So we are collaborating with these groups, and we are including our patients here in Toronto that are diagnosed with EHE into this PUSH platform to have a database going forward with patients diagnosed with EHE. And we're doing this with multiple different groups across North America and Europe. So our third aim is to create clinically annotated EHE PDX models. So what does that mean? So that means that we are using biopsies of tumours of people with EHE in Toronto with their consent. We are going to do this moving forward. We haven't started this yet, to develop tumours and grow tumours of EHE in the lab that we can then test treatments on to develop some treatments for patients in the future. And then the fourth aim is a preclinical evaluation of mTOR inhibitors. So mTOR inhibitor example would be sirolimus. And we are doing this in collaboration with Dr. Kristianne Allen's lab in the University of Toronto. So she is using different techniques to try different treatments, including mTOR inhibitors, on EHE tumours in the lab, and we are having some good results with this, and this is still ongoing, and we are hoping to publish this data in the future. So I'd like to thank you for your attention, and then again thank the organisational committee for inviting me to present on our research today. I'd like to thank our co-authors and all of these other hospitals that have made this project possible, and also the Sarcoma Research Foundation of Canada for giving us the funding for this.
Yep, we'll see you back in the Q&A session. Our second presenter in this session is Dr. Gujral, who's going to talk about TRACER and breaking barriers in rare cancer.
All right, thank you for this invitation to present here at this EHE 360. So I'm going to step back and talk more broadly about rare cancers. Rare cancers as a challenge, and what are the challenges, and how we're coming together as a community as an initiative that we started about 2 years ago, actually 2 years and 1 month. It's called Transformative Rare Cancer Initiative here in Pacific Northwest, coming out of several different labs. So there are more than 20 labs that are studying various different types of rare cancers. From Hutch Cancer Center, Seattle Children, University of Washington, Institute of Systems Biology, and forging this community that comes together and build resources that can help build shared resources that could be shared with the community. So very briefly, rare cancers, by definition, is defined in the US as any cancer with less than 15 incidents per 100,000 people. In the US, a little bit more stringent, it's less than 6 per 100,000. Individually rare, but collectively they can make up to 1 in 4 cancers worldwide. Now, studying even most common cancer is challenging because of the heterogeneity and complexity that is observed in the human tumors. Now, imagine all of those challenges are severely amplified in rare cancers, where you have fewer patients, Fewer patients means fewer samples. Fewer samples means there are fewer models for us to study in the lab. That also means there's a lack of molecular understanding of what are the critical drivers, what are the potential therapeutic targets. That is also connected with lack of therapeutic options. So this is the rare cancer recognized as a major unmet medical need and sort of the landscape that we hope to change by coming together as a community that addresses these central challenges in studying rare cancers. So our vision is to bring together the community that includes lab scientists, include clinicians, oncologists, surgeons, patient advocates, come together and address some of these challenges. We have three sort of main focus areas. The first one is something I alluded to earlier, is the lack of preclinical models. As we heard in many of the presentations today, there are preclinical models which are used in the lab. So it could be 2D cell lines, it could be organoid, it could be patient-derived xenograft or PDX models. So just by the virtue of having fewer cases, there's also a lack of preclinical models for many rare cancers, including EHE. And the second area that we're working on is also the sparse molecular data. So it's glad to see that some of the presentations that is changing in the EHE. But in general, most rare cancer patients do not undergo transcriptomic or exomic profiling. So there's also lack of molecular data across many rare cancers. And the third challenge that we're hoping to address is also how to translate some of these lab-based discoveries into clinic. Running a clinical trial in a rare cancer is a challenge. So this is also something that we are hoping to address through new mechanisms from the FDA, is how to translate discoveries from the lab into clinical space. So our main work engine is this sort of a cartoon that illustrates a tracer bioresource. So we work collaboratively with biobanks internationally and also locally with the pathologists and oncologists at various different cancer centers. Our goal is to be able to bring a fresh tissue to our lab within 24 to 48 hours from anywhere in the US. And this tissue is then banked and with extracted with molecular information, so DNA, RNAs. Every sample that we get, we do omics profiling. So we also try to generate preclinical models from these fresh tissue. That includes cell lines, includes something I will actually later on describe called microtumor models, as well as patient-derived xenograft models. So in the past 2 years, we have collected over 300 cases of rare tumors, representing from over 40 different types of rare cancers. And of these, we have been able to cryopreserve about 112 rare cancer cases. So that includes 5 EHE cases. I know, most biobanks traditionally, they would either drop the sample in a formalin or snap freeze, which is great for long-term storage. But when you're trying to generate preclinical models or do functional testing, they fall short. So this is one of the things we're hoping to also share our protocols on how to viably cryopreserve a small chunk of tissue, not dissociated cells in single cells, but as a tissues, which can later on be used for functional testing. They can later on be used for generating preclinical models. The entire piece of tissue could be implanted in mice to develop PDX in the future. So that's sort of the living biobank that we're generating. And so far, we do have 5 EHE cases that we are sharing with other members of the community here. We've also established, using these microtumors, we have made cell lines and PDX models. So just to show you the landscape of roughly about 3 tumor samples that we processed in the past 2 years, three samples per week. And most of them are in the GI space. That's the focus of my lab, and some also into sarcoma and CNS space. For many of these cases, we have biobanked biomaterials, DNA, RNA, FFPE, and as something I mentioned earlier, is a microtumor model. That's sort of a workhorse model in the lab that allows us to make new models in the future and also do functional drug testing directly on these 3D pieces of tissues. So what can we do with this data? Just show you a landscape of looking at EHE. As I mentioned earlier, so the EHE cases that we have, they are all from liver. So then we could ask a question, okay, the EHE that occurs in the liver, how does that actually compare with any other cancers that occur in the liver? So in the liver, the most common cancer is hepatocellular carcinoma, shown here in this red circles. So this is a dimensionality reduction map of over I think 600 liver tumors. And if you put them in a two-dimensional space, you will see the HCC sort of cluster together. And all of these rare liver tumors that also occur in the liver cluster separately from HCC. And you see here EHE, we have 11 cases here, 5 from our database, as well as 6 from the publicly available data. So we could put all this data together, harmonize it so we can start to look at it and start to compare differences between cancer types and compare difference between EHE and a normal tissue. You will see here EHE forms a distinct cluster, but it does also overlap with another tumor that occurs in the liver that we have here is angiosarcoma. So just since it molecularly it's a distinct cluster, which also suggests that the drugs that are effective in HCC or cholangiocarcinoma may not work in EHE because it's the underlying biological processes that drive EHE are very distinct. Shown here is something that you can see that Ty1 is a marker for endothelial cells. Now it is very low in the non-tumor liver, also low in many of the other liver tumors, but it's quite high, which makes sense from this cell type of origin of EHE, as well as angiosarcoma, which share a similar cell type of origin. The other thing we can see is, okay, does the data actually make sense? So we look at the expression of CAMTA1 and W2TR1 TAS. And again, there's an increased expression or a higher expression that we see compared to any other tumors that exist in the liver and also non-tumor liver. So many of the cases that we have the EHE, we also have a matched non-tumor liver from the same patient. That also gives us sort of better quality data to be able to compare the same patient non-tumor liver as well as the EHE tumors. So this is, just having this data set, it's great for a community, and they can actually use to ask questions about what makes EHE unique and what are the underlying pathways and processes, and how is it distinct from other liver tumors. So I'll show you a couple more examples. So one of the things we looked at is, I know this has been a central focus of many different talks today, is the mTOR pathway. So mTOR pathway, in general, it is higher in tumor compared to non-tumor tissue, as seen here. But you see, compared to the other tumors that occur in the liver, EHE does stand out. Angiosarcoma as well, where we only had 2 cases, but here looking at 11 cases, the mTOR pathways increased, the activity of mTOR pathways increased. Now, another thing you notice that there's also a great degree of variability among patient to patient as well. So there are some patients where do see a higher activation of mTOR pathway compared to the others, but overall, this activity is higher compared to non-tumor liver and also compared to other common HCC as well as some other rare liver tumors. So next thing we asked is, so the FDA has like over 300 small molecule inhibitors that are approved, many for oncology and also many biologics. And the biologics are these antibodies or antibody drug conjugates that are approved for various other cancers. So these are antibodies that are attached to a cytotoxic drug. So antibodies sometimes used as a homing device, and they recognize a receptor on a cancer cell or a surrounding cell and deliver the cargo, and that ends up being a cytotoxic for cancer cells. So these are cell surface receptors. So the questions we ask is, are there any unique cell surface receptors or our cell surface receptor for which we may already have ADCs or antibodies that could be readily repurposed? So we looked at, compared the expression of these cell surface receptors. Again, this is a cell surface expression, the mRNA level, which might be different from the protein or needs to be validated or confirmed with the protein level. What we found is that compared to non-tumor liver from the same patient or from a group of patients, EHE showed a much higher expression of TROP2 or TAX-TD2, Nectin-4, and few other few other proteins. Now, for each of these markers that I'm showing here, there's a antibody or antibody drug conjugate that is already approved by FDA for other cancers. Just to show you here as a TROP2, which is very low level in the non-tumor liver, low level in other HCC where it's not approved. But we do see actually quite high expression level of TROP2 in the EHE. And this expression level is also highly variable. So at least more than 50% of patients, that's a very high level of TROP2. And TROP2 ADC is approved for triple-negative breast cancer and as well as bladder cancer. Again, so using this kind of data allow you to generate the hypothesis that could those hypotheses could be tested in preclinical models, could also be tested in patient tissue directly using these TROP2 ADC to see if there's, and again, if as we build this database and expand to a more number of patients, that also helps us to hone our hypothesis whether this is a really a marker that could be exploited in EHE, or is this sort of very patient-specific signal? So coming back to the microtumor models that I alluded to earlier, that it's very important to cryopreserve these 3D tissues. And the reason I say that is, as a cancer biologist for past 70 or 80 years, we learned a lot by making cell lines. So when we get a tumor sample, we break it apart into single cells. We put them in petri dish. We learn a lot about their behavior. We learn how they grow, how they proliferate. We learn very fundamental biological mechanisms like cell cycles, cell migration on petri dishes. That has taught us a lot. But when it comes to testing whether those drugs will be effective in killing cancer cells, we do a great job on petri dish, but those same drugs are somewhat not effective in vivo when cells are surrounded by other cell types. So that's why our lab has put a lot of effort in generating these 3D microtumor models, which are different from 2D culture, also different from organoids that can be made on the same day as we get a fresh tissue. So when you get a fresh tissue, something actually we learned from neuroscience. So where they have been developing called hippocampal brain slices from the 1960s. So when you get a fresh tissue sample, it could be a cold needle biopsy or it could be a large surgical resection. We cut into about 400 micron slice. These slices are then mechanically dissociated into these small tumors, what we call a microtumor, as seen here at the EHE. And these microtumors are 3D pieces of tissue, 400 micron thick. They contain multiple cell types. They contain native extracellular matrix. There's no matrigel, and they could be cultured viably in a lab for at least a week and sometimes even much longer. And shown here, so you can see the staining of the T cells, staining of endothelial cells, showing the strongest and the highest staining of all the other liver tumors. That makes sense from the cell type of origin. Also showing some macrophages and pan-thyrocyte marker in EHE. So these microtumors can be viably cryopreserved so that in future you could actually bring them and evaluate drugs on them, or even use them to make other models like PDX models or cell lines. So I'll show you like how we're using these microtumor models. So we have a platform which is now a CLIA approved platform, what we call a smart match. So the idea is that we bring a tumor samples that comes to our lab from fresh surgical resection. We cut them into slices, from slices or needle biopsy, cut them into these microtumors. These microtumors could then be used directly for drug testing. So we found that drugs that a recent study that we published last year where we contrasted a drug screen on a 2D culture and these 3D microtumors. So we took a cancer cells, actually a series of cancer cells, mouse cancer cells, and treated them with several hundred drugs in 2D culture. And we put the same cells in mice where they develop tumors, and from those tumors, we made these microtumors and did the same drug screen. What we found was there were 3 times more number of drugs that were effective in 3D microtumors than in 2D culture. It turns out, and we followed up on some of those drugs, and they did, in fact, work effectively in vivo. So what we found that many of those drugs were targeting non-tumor cells, so they were targeting cancers in fibroblasts or immune cells or endothelial cells, which were not present in 2D cells. So which also suggests that our traditional 2D screening tend to miss out on some effective drugs that could be potentially effective in 3D microenvironment as well as in vivo. So this is our workhorse model is using these 3D microtumors for drug testing. Now, the amount of tissue that come from the OR for any surgical resection biopsy is highly variable. So a lot of time patients are pre-treated with chemotherapy or radiation or other forms of therapy. So there may be a lot of necrotic tissue. So it's not feasible to do a large-scale screening in these 3D microtumors. Which is actually why we make 2D cell lines that allow us to expand those cancer cells in culture so that we can test hundreds or thousands of drugs. So this is where actually our lab has integrated some form of machine learning that allow us to do a large-scale screening even in 3D microtumor. So our idea here is that we screen a small number of drugs, about 50 to 100 on 3D microtumors, which we can. And then we use the result from these 50 to 100 drugs and then use a machine learning model that can predict what would be the more effective drugs from the panel that we have not tested. So one way we can address this challenge of limited material for drug testing is by doing an iterative approach. So testing a few drugs with a known mechanism of action and using the results from this, we then build a model that would predict what would be the effective combination or what would be the effective drugs. And the nice thing is the entire process is done within 7 days, so we are able to report back to the oncologist. This is sort of a rank order list of drugs that are effective in 3D microtumor for this patient. So we've applied the smart match approach in about 80 cases now. This is a bit outdated slide, and that includes actually 2 EHE cases as well from the liver where we have done screening. So both of these patients had undergone a liver surgical resection where we did the screening. And we have actually drugs that were effective for both of these patients. However, the patient has no evidence of disease, which is great. But in case this tumor does recur, that would also be an opportunity for us to evaluate whether the drugs that we found ex vivo would be relevant in vivo or would actually work in the patient. So just show you sort of one example. We have this Ewing sarcoma case from a 17-year-old from Seattle Children who had gone through 5 lines of therapy, mostly toxic chemotherapy or PARP inhibitors, and had failed all of those progressive therapies. So when we got a surgical resection, we did our screen, we were able to predict responses to about 1,000 drugs. And of those, because we have cryopreserved tissue, so we also are able to test our machine learning based prediction on the tissue, and we report the experimental value shown here in orange bars. And the drug that we found was most effective in inhibiting the viability of the 3D microtumor is avapritinib. Now, avapritinib is not approved for Ewing sarcoma. It's actually approved for gastrointestinal stromal GIST. It is an inhibitor that targets a cell surface receptor called c-KIT. So when we asked, does this case express c-KIT? So looking at our c-KIT expression across the cohort of our cases, again, this GIST or gastrointestinal stromal tumor express high level of GIST, which makes sense because avapritinib is approved there. And this particular case, and also 2 other Ewing sarcoma cases, also express high level of KIT, but not as high as GIST, but does express overexpress KIT. And so because of this reason, this drug was actually then chosen by oncologists, and the patient who had before to this regimen had many complications, required hospitalization for constant IV management, for constant pain management. And since the avapritinib has been actually done very well, and the pain has been managed, and been alive for 6 months, and the scans are pending. So again, this is a non-obvious target that we could not have predicted that avapritinib would be effective in Ewing sarcoma. Now we are sort of expanding this approach to looking at other Ewing sarcomas that may have a high expression of c-KIT that could open up opportunities to repurpose this drug from GIST into Ewing sarcoma, especially for patients that have high expression of c-KIT or activation of c-KIT pathway. So again, this sort of what we call a functional precision medicine, which is directly testing drugs on patient tissue, is complementary to genomic approaches. So it's complementary to molecular analysis. So we have recently started a clinical study at the NCI with Dr. Jing Wu. This is our NCT number, actually about 2 months ago. And the idea here is we take a CNS or the brain tumor patient who have exhausted other therapies. So they have gone to radiation, temozolomide, and several other therapies. And if they're having surgery at the NCI, their surgical resection tumor is sent to us at Hutch. We would do the SmartMat testing and report the results back to Dr. Wu and other oncologists who would consider it together with molecular profile and other data. And the idea is, this is to be done over like 80 to 100 patients. So the idea is that we would look at our screening results ex vivo, where we can test hundreds or up to 1,000 drugs ex vivo, and identify patients who are responding to certain kind of drugs, and also be able to link what are the features or the molecular features of these GBM patients that are responding to certain type of therapy, to be able to design a better clinical trials in the future. So we're doing something similar, as I showed you a case from Seattle Children, which is all pediatric cancers or rare cancers, with Dr. Shireen Ganapathy. as well as rare gynecological cancers with Dr. Banda at Fred Hutch Cancer Center. Since this is a clear approved assay, we're hoping that this could be expanded not only to GBM or pediatric or rare gynecological, but every rare cancer, including hard to treat cancers that have exhausted other therapies. Again, something I had previously mentioned, we don't see this as a new thing. It's actually sort of something that's complementary to genomic profiling and is very much relevant in rare cancers where we don't have potentially actionable markers that could be linked to a existing drug. So direct functional testing can allow us to uncover some specific vulnerabilities for rare cancer. So I showed you an example of avapritinib, so c-KIT expression in urine. So these are some of the findings from functional testing that could be then exploited in designing better clinical trials that will combine both genomic profiling as well as functional precision oncology. Then coming back to our key initiatives, we've been around for about 2 years. We've been able to collect about roughly 3 cases every week from various parts of the country and established a very strong proponent of creating a living biobank, especially with microtumor models. And using these microtumor models, we can generate other preclinical models, as well as every case that we get also undergoes the multi-omic DNA RNA profiling. So it's not only important that we understand that we get some drugs that are effective, but also understand why the drugs are effective so they can be used in other patients that may have a similar genetic alterations in the future. We've opened 1 clinical study at the NCI and hoping to launch a few more. And this is a clear lab assay, which means the results could be shared back with the clinicians with potential application for patients' treatment. With this, I just want to thank some of the members of my team. Kristi Sherman is a program manager, and the SparkMatch team is led by Dr. Marina Chan and Dr. Song Lee, as well as Deanna. Also advisor of our tracer program, Dr. Eric Holland. Also members of the rare cancer community, John Hopper, Jim Palma, who's the CEO of Target Cancer Foundation, as well as the oncologists and surgeons, Dr. Brooke Hovitt from Stanford, as Dr. Jing Wu, who's running a clinical study at the NCI. This will come back at the Q&A session, and here's our website and my email. If you have any other questions that's outside of this presentation, please feel free to reach out to us. Thank you.
Thank you much for the very informative presentation. We'll see you in the Q&A session, and I turn it over for the last presentation of the day to EHE Foundation's own Kristi.
Thank you, David. And thank you so much to Dr. Gujral and Dr. Weadick for all of your incredible work and for sharing it with EHE community. I'm going to be as short and sweet as possible today. As you all know, my name is Kristi. I'm the Director of Research and Engagement here, and I'm going to take just a few minutes here at the end of the day to talk a bit about some ways that people with EHE can raise their hand and contribute to research. We've just heard some amazing examples of how knowledge is growing from the collective input of many people with EHE. And you may be asking yourself, what about my journey, my tissue, my experience? Does that really make a difference somehow? And what is the true value of one person in basic science research and clinical trials for EHE? Those experiences and tissues and specimens and all of the treatments you've tried or not tried over the years, those are all really important data to scientists and clinicians who study EHE. The research and development industry globally is built around success in diseases like lung cancer and breast cancer, those big cancers which have large patient populations and clinical trials that enroll thousands of people. In ultra-rare sarcoma like EHE, we're navigating the same system as the big cancers, but with extremely small and often even unknown numbers of patients. So our clinical trials have a handful or maybe 50 people at most. The groups of people that are being compared in trials for common cancers are very well defined, and we know the natural history of their diseases, while EHE studies often include people with very different presentations that affect multiple organ sites. When measuring success, the big cancers have known success criteria, many of you have heard of RECIST, and very clear benchmarks for improvement, while in EHE, success and improvement are much more individual. Even before a drug makes it to a clinical trial, researchers studying those big cancers have a lot of tools to study and learn from, while in EHE, we have precious few that don't currently represent the full spectrum of EHE. Because of this David and Goliath scenario, the EHE research community has some unique challenges, but it also means that each individual has a uniquely big impact on the EHE research landscape as a bigger part of a smaller whole. Researchers have to carefully ask key questions. How does a treatment work in people with EHE? What are the biological differences between groups? And how many people actually have EHE, and how are they different from each other and other cancers? In particular, drug companies often come to us to understand how many people could potentially benefit from a new therapy and what that benefit actually looks like. It turns out that that isn't a question with a clear, straightforward answer. As an example, let's say we have 10 people with EHE, but of those 10, 7 have metastatic disease, 5 have EHE in their lungs, and 2 have it in their bones. 6 have EHE in the liver, 4 do not. 2 people have had liver transplants, 6 have had surgery, and 3 have tried some kind of systemic therapy. Suddenly, this isn't just 10 people with EHE, it's 10 very unique and individual presentations of EHE that are similar and different to each other in very specific ways. So when we ask, as the research community, what works for you, we're also asking, what is your EHE like? What is your biology like? Who is similar to you and in what ways? Each individual experience helps scientists and researchers understand how and why a treatment might work better for some people and not for others, and who can really benefit. So as an individual living with or caring for someone living with EHE, your experience is immensely valuable, and you have an opportunity to bridge the gap and contribute that information to the scientific community in a couple of ways. The first opportunity is the EHE Biobank, which we've already seen in action in this conference. If you remember earlier today, Dr. Duane and Dr. Tanas's lab shared data that was only made possible through the EHE Biobank, when donated tissue was turned into cell lines that were used in experiments to answer research questions. But just like the people they come from, cells and tissues are all just a little bit different from person to person, and we need more people that represent all of those different small groups of EHE to donate their tissue too. You can see our very own Maggie here in this picture with one of our collection kits. She volunteered to share this picture with you all today. It's very simple. I encourage you, if you're interested, to scan the QR code and you can learn more about what the process is like. You don't have to have a plan to have a surgery or procedure scheduled right now. You can get connected with us and we can help you make that plan. For people living outside the United States, we will also help connect you with our international partners who are doing similar work. Another opportunity to turn your journey into real data is the EHE Global Patient Registry, which many of you may have signed up for, but which I encourage you to explore if you have not. The goal of the registry is to describe experiences of people living with EHE over time, treatments and outcomes from those treatments, symptoms and how they change and impact quality of life, and even inform overall care guidelines or identify people who might be candidates for clinical trials as they develop. The registry is a natural history study of EHE, and it's one way that we at the Foundation can take all of your experiences out there in the big wide world and translate them into real usable data that scientists and researchers can learn from. That truly is one of our greatest roles here at the Foundation, to collect all of those individual narratives, specimens, and tissues and translate those lived experiences into data that is actually usable for the scientific and medical community. Your experience is incredibly valuable, but it must be collected in a structured and intentional way in order to be interpreted and learned from, in order to improve outcomes in the future. So if you take one message away from this conversation, I hope that you hear me when I say an EHE one is never just one. Every experience, every journey, every person matters that much more when learning about a rare disease like EHE. One person could be the key to understanding something brand new about EHE, and that person could very well be you.
Thank you much. And let's gather back our session 3 participants for our final Q&A. And first question I had, Dr. Weadick, like the temporal information you collected is amazing. And you noted in the data set there, some statistics on sort of the genetic testing and a positive trend line there. I'm curious if you notice any sort of trend lines in the types of treatments people are getting. We saw sort of the collection of treatments for the big group. If you know, were there sort of trend lines, or were we seeing any newer things in the more modern years compared to the older years?
So I don't have the exact data of that to hand, but I do know that we did see more use of mTOR inhibitors more recently, particularly in the last kind of 5 to 10 years, and more of the doxorubicins and the more toxic chemotherapies in the earlier years. But yeah, that's what we've seen.
That's good news. I mean, right, I think both on the genetics and the treatment, then we're seeing clinicians wherever they are, right? Because you've got all this data from a broad swath, are hearing about the research and the information's traveling.
Yeah, and maybe thinking about more of what they're treating and not just giving the chemotherapy, because that's all that they have. So yeah.
Oh, excellent.
I'm actually going to take a question that I can answer, which is wonderful. Someone asked if you can donate tissue from a previous biopsy or if the tissue has to be collected at the time of the procedure. And certainly I can't speak to the collection procedures in Dr. Gujral's lab, but at the EHE Biobank, we can take a variety of kinds of tissues. It may be used for different types of research depending on when the tissue was collected and how it was preserved. So there's lots of different ways that tissue can be used in research, and depending on how it's collected, whether it's collected, before or after or with the intention of research being done, we can use them for different purposes. And Dr. Gujral, if you wanted to elaborate on that, you're more than welcome to do so.
Thank you. Absolutely. So if the tissue was collected previously and is fixed, if you have FFPE or some blocks that were made, we'd be happy to take a small part of that tissue, bring to our biobank. And what we would do is a sort of very extensive molecular profiling. So that data then goes back to this two-dimensional map to see how this case, how a patient actually compares with other EHE cases. But if it's a fresh tissue that we can potentially use it for preclinical models, then it needs to be sort of prospectively collected. But historically collected tissue, if they're frozen, if they're fixed, they could still be useful. It could be useful collecting molecular data, but they will not be ideal for making functional testing or making preclinical models.
I have a question, Dr. Gujral, based on your presentation. It was very interesting to see obviously the breadth of what you're doing with all of the rare cancers, but then some of the specific details you provided on EHE. And in the one presentation, you'd identified a few potential interesting markers that could be therapeutic targets, like the TROP2 and the Nectin-4. And I'm curious, is that something your team is following up on, or is that, do you have any thoughts about those as sort of shots on goals that might be interesting for someone to follow up on?
Absolutely. So, there are ADCs that are FDA approved for both of those, and there are actually even clinical trials to expand labels on those. So there's a lot of opportunities. But one thing, as I mentioned, our samples were liver specific. And what Kristi showed, the EHE could occur anywhere else. So one thing before we sort of jump the gun, would ideally to actually sort of validate whether this is a bona fide marker to go after, is to look at other EHE cases. This could be done by immunohistochemistry across, a panel of EHEs in a biobank or other sources. And if it turns out that EHEs do express high levels of TROP2, and I think there's definitely an opportunity here to even do a combination therapies with the TROP2 ADC and perhaps with other forms of therapy. But again, first thing would be to just do IHC across a large number of EHE from various different sites to confirm that these are potentially actionable markers. And we have a couple that we would do IHC in our panel, but this is something we would love to work with the community who have other samples from other sites, and then maybe we can sort of combine forces and to look at the cell surface expression of already approved ADCs and biologics, and help and come with sort of informed strategy of what would be the next step forward.
Excellent, thank you.
Thank you. I'm going to actually ask Denise if she can join us too. I'm very cognizant that everyone has donated quite a bit of their time to this event today. And I'm going to pass it over to Denise to wrap us up for the afternoon.
Thank you. Thanks, everybody. Thank you, Dr. Weadick, Dr. Gujral, David, Kristi. Great job. Lots of learning today, truly. Just a lot of learning. The questions and the answers that were provided were great. I really we all appreciate the time and the information that you pulled together to share with us. And I think that, the takeaway is there's a lot of momentum. There's a lot of new data that has come out. I think that just the collaboration that we're seeing around the world is wonderful. So thank you.